Hello friends, in this video we are going to be discussing briefly about secondary growth in plants. Like we always study a pre-log to whatever we are going to discuss further. In secondary growth of plants, and to understand it, you must know the organization of plant tissues, which we study in plant anatomy, right? We know that plant tissues are either meristematic or permanent. Meristematic are the undifferentiated tissues which have the power to divide, and permanent are the differentiated tissues which have certain fixed shape, size, function and property, and they do not have the property to divide, right? The meristematic tissues are further classification on the basis of their location and their origin. So we say those meristematic tissues which are present in the embryonic tissue are known as the primary meristem, which are organized in the center of the plant tissue. There is a procambium which is going to be the precursor of the vascular tissues. Surrounding them we will have peribelum, the precursor of the ground tissue, and outermost layer of the primary meristem in the embryo is known as the dermatogen, which is a precursor of the epidermal tissue. When this embryo grows, it shows differentiation. For this you need to know the physiology of plants, that every plant cell shows certain sequence of growth and development, right? Growth means increase in number, development means changes from the time it is born till the time it dies, right? And the developmental changes include differentiation, right? The first thing that it does post being formed, it grows in size and it loses the power to divide and attains a permanent function, and now it is known as a permanent tissue, which are organized into certain layers in the plant tissue, which are known as the tissue systems. The innermost is the vascular tissue system, which contains the permanent tissues that are complex nature, xylem and phloem. The second, which is surrounding the vascular tissue, is the ground tissue system, which is majorly made up of parenchymatous tissue or sclerenchymatous tissue. Outside on the epidermal tissue system, I have the protective sclerenchymatous or the tensile strength collenchymatous tissue. So they are all are permanent tissues which are organizing in the transverse section of the plant body. If I cut a plant body and I observe the transverse section, all the tissues which are occupying the space are permanent. In these permanent tissue, between the permanent tissue, I have certain meristematic tissue which are not present in the embryo. These meristematic tissue which are present between the permanent tissues are known as the secondary meristem or the lateral meristematic tissue, which is responsible for secondary growth. So we say the secondary growth in plants, the tissue responsible is the lateral meristematic tissue, which is a secondary meristem that occupies a space between the permanent tissues of the tissue systems and is known as the secondary meristem, is responsible for secondary growth and is specifically seen only in dicots, dicot root or dicot stem. So you will not see any secondary growth in monocot root or monocot stem. There is no secondary growth in leaves, so we don't talk about it. Then when it is present between the permanent tissues, it can be present between the permanent tissues of vascular tissue system, it can be present between the permanent tissues of ground tissue system, or for that matter epidermal. We observe them in two tissue systems, that is vascular and ground. When it is present in the vascular tissue system, that lateral meristem is known as vascular cambium. And where is it present? It is present between the xylem and the phloem as a vascular cambium ring. So this is the vascular tissue system, this is ground, this is epidermal. In vascular tissue system, this vascular cambium is present between the phloem outside and the xylem inside as a vascular cambium ring. Likewise, when it is present in the ground tissue system, it is known as cork cambium. That's the site where it is going to be present in the vascular or the ground tissue system, which we see as here, that if this is the vascular tissue, this is the ground tissue, the blue one, and the red one is the epidermal tissue. Then in the ground tissue, just below the epidermis, there is a layer of cells which forms the cork cambium or the lateral meristematic tissue of the cortex. This lateral meristematic tissue is secondary meristem because it is formed post embryonic development. And also to be noted, this cork cambium is formed post there is secondary growth in vascular tissue. Once the vascular tissue has shown secondary growth, form the secondary vascular tissue, that is secondary xylem and secondary phloem, then it activates certain cells below the epidermis of the cortex to become de-differentiated. What do we mean by de-differentiation? That's the physiology involved in secondary growth. That the permanent cells lose their specific property, the differentiated property, and gain the power to divide again and form secondary tissue. This secondary tissue is what we call as a secondary growth. So once there is secondary growth in the vascular tissue, the cortical tissue which is below the epidermis gets activated, shows de-differentiation and becomes meristematic again to divide again and form new tissue. Now this new tissue is not the primary tissue formed post embryonic development, it's a secondary tissue formed. That's why we name it a secondary tissue and we name it the secondary growth, right? Once the new tissue is formed, these new cells again undergo the developmental changes, they again become differentiated, that's why we call it the re-differentiation to perform a specific function. So first there will be de-differentiation, formation of the cambial tissue. Then there will be re-differentiation, the cambial tissue which is leading to the formation of new cells is again going to become specific in its shape, size and function and perform a specific function. Clear? What is the end result? The end result is the formation of the secondary tissue. Like if I have vascular cambium ring which is present between xylem and phloem, this vascular cambium ring divides and forms cells towards the inside as well as towards the outside. When it divides towards the inside, it is going to form secondary xylem. When it is going to divide towards outside, it is going to form secondary phloem. So the secondary phloem is going to crush the primary phloem. So in the process of secondary growth, the primary phloem gets crushed. And when the secondary xylem is forms the bulk of the plant body, it is known as the wood. And when the secondary xylem grows, it also crushes the secondary phloem. So phloem tissue is very, very small in amount in a mature plant and we known to call it as bast or the bast fibers of the phloem fibers. Once there is secondary growth in the vascular tissue, the inner part has grown. So what is growing? The diameter, because now it is present in the form of a ring. So when the ring expands, it expands the diameter. That is why we say that the lateral meristematic tissue or the secondary growth is responsible for the increase in the girth of the plant body, right? Once the internal tissue system growth increases, it gives signals and activates the tissues which are present in the cortex or the ground tissue system just below the epidermis, and those cells become de-differentiated, form the cork cambium, the green line. This cork cambium is also known as phellogen. It divides on the inside, forms a secondary cortex tissue which is known as phelloderm. It also divides on the outside. When it divides on the outside, as you can see, it breaks the epidermis and it produces discontinuities in the epidermis. And those discontinuities appear as pores called lenticels, which are responsible for exchange of gases on the surface of stems or roots for that matter, right? This outer layer is known as phellum, also known as cork, my dear. So all the three layers together, phellum, phellogen and phelloderm, they are formed in the secondary growth of the cortical tissue which surround the secondary vascular tissue. So we say all the tissue that is outside secondary vascular tissue that is formed by secondary growth in the cortex, including the phelloderm, phellum and phellogen, is known as the bark. So anatomically, bark contains of three tissue layers in a plant body: phellum, phellogen and phelloderm. All the anatomical tissues which are present outside the secondary vascular tissue are considered under the bark. The outermost layer of the bark, that is the phellum, the lastmost, is known as the cork. This is the end result of the secondary growth in plant body, be it stem or root, usually seen in dicots only because dicots have lateral meristematic tissue which is going to be responsible for secondary growth and increase in the girth of the plant in two tissue systems. either the vascular tissue system leading to the formation of secondary xylem or phloem, or the ground tissue system leading to the production of phloem or phloderm and forming the bark of the plant body. Yeah, just giving you a brief outlay of secondary growth. Hope it is clear to you. Hope to see you soon again with more videos and increasing your knowledge in biology. Thank you so much.